Okay, greetings students. A warm welcome to this session of the Algorithmic Thinking in Bioinformatics course. I'm actually quite excited for today's session because this is going to be our very first real world problem session. So we are going to start with a real world biological question, a very interesting question related to how a cell senses what is happening outside the cell. How does a cell sense its environment? And then we are going to address a computational solution to this problem. We are going to see how the power of computation can be used to figure out the different routes through which a cell senses its environment and then relays that information from the outside of the cell to the inside, to the nucleus of the cell. And another interesting component about this particular session is that the computational solution is going to naturally involve concepts from two different areas in computer science. One is the graph algorithms, a randomized graph algorithm that is based on dynamic programming, and another is a machine learning model that is based on logistic regression. Okay, and both of these concepts are going to be spontaneously or in some sense naturally involved in addressing this particular question. Okay, so without much further ado, let's get started. In terms of the context for this particular session, let's look very briefly at what we have seen so far in the course and then what this particular session will be about. So what we have seen so far in the course are essentially two sets of sessions. One are the introductory sessions where we have motivated this course from various perspectives, essentially arguing that bioinformatics or computational biology provides a set of real world problems that helps you practice end-to-end data science, right? And then we have provided a background session where we have introduced some necessary glossary to understand the biomolecules and cells that are part of life, right? And so that we will be able to connect the language of life to that of the computer. Okay. So that for instance, you may recall that a gene regulatory network is a biological concept on how genes regulate each other through a network or circuitry of relations among the genes, and how that can be represented as a graph using the adjacency matrix or adjacency list representation of a graph, right? And today we are going to talk about an informative biological question, right? So we are going to follow the course style in terms of this active learning style, where we pose a biological question and then we try to answer or formulate a computational problem to address that question and then show some solutions for that computational problem, right? And that is what we are going to see in the context of one particular question, which is how does a cell sense and relay information from its outside to inside. That is why I call it an informative question, pun intended. So the cell has a lot of information that is happening outside the cell, and so it has to sense that information and then it has to relay it to the inside of the cell, specifically to the nucleus. What are the molecular or signaling pathways or routes that are involved in this sensing and relaying of information from the outside of the cell to the inside of the cell? So given that this is the biological question we are interested in solving, how would you understand whether computation has even any role in solving this biological question, right? So we need to make sure that we have a good idea of the computational problem that could be used to solve this or address this biological question. And one particular such computational problem could be, can we find all possible routes or signaling pathways in a large scale that are hidden in a large scale biological network? There could be some high throughput experiments which have given you a large scale biological network, and then these signaling pathways could be hidden as substructures within these large networks. So can we find these substructures, these signaling pathways that are hidden inside a larger biological network? So that could be a good starting point for a computational problem. We still need to make it much more concrete. We need to define what a network is, what a large scale network is, what a substructure is, and so on. But then that gives you, but this kind of description or this kind of question on can we find all possible signaling pathways hidden in a large biological network would be a good starting point for an associated computational problem. Once we have this computational problem concretely formulated, then we can think about algorithmic solutions, and in this particular case we are going to see that there is a natural way in which two solutions come together. One is an ML method, a machine learning method, and another is a dynamic programming based algorithm called color coding algorithm. So we are going to take this machine learning classification method and a colorful solution, and both of them are going to contribute to the solution to answer this particular question on how a cell senses external information. So, let us just start with the biological question. So, what are the different routes a cell takes to sense and relay information from its exterior to the interior, right? And before we delve into this question a little bit further, right, or deeper, let us answer an even more basic question. Why does a cell have to sense its environment, right? And to those who have studied cells, who have looked at it inside a microscope, for whom it is their day job to understand cells, the answer is actually quite obvious. But for those who are studying cells for the first time, just think about it, pause and think about it, right? A cell cannot perform even its very basic function, or even whether to decide, even decide whether to survive or grow or die without knowing what is happening outside the cell, right? And let us take an example of the body's defense cells or immune cells, right? The immune cells has to identify the sources of infection, right, and then it will have to go towards, migrate towards those sources of infection, and then have to act upon the bacteria or virus or whatever other pathogen is there, right, causing the infection. So, it cannot do this job if it does not know what is outside the cell, because it needs to constantly monitor what is happening outside the cell to figure out where the molecules that are being shed by these bacteria are coming from, and then it has to migrate towards that region, right. So, in this case, the body's defense cells cannot perform its basic function if it does not know what is outside it. And there are some other examples like fat cells and cells that are deciding whether to grow or die. They also need to be aware of what is happening outside the cell's membrane, right, in order to decide whether to a fat cell needs to decide whether to store energy or release energy, right. So, it has to monitor what is happening in the blood vessels that are surrounding the fat cells, and that blood vessels carry certain signaling molecules or hormones such as insulin and certain other molecules such as glucose, and the levels of insulin and glucose will determine whether the fat cell needs to store the extra glucose or needs to release the energy that it already has in its stores, right. Similarly, a cell can decide whether to grow and divide into daughter cells, or whether it wants to actually commit to a programmed cell death based on some external signal it receives. Okay. In multicellular organisms, cells are often sending signals to other cells, and so a cell always need to be aware of the context in which it is present and what are the external signals that other cells are providing and what is the environment telling. And so, it has to keep on doing real time monitoring of its environment to make real time decisions. Okay. So, it is very important for a cell to sense its environments and to relay it to the interior of the cell. And all of these words that I just said, a video or a picture is worth 1,000 words. So, you can see here that this large blob is the neutrophil, okay, and then this little black dot that the neutrophil is chasing is the bacteria. Okay. I am going to leave it as a suspense to know whether the neutrophil, our body's immune cell, is going to eventually catch the bacteria or not. These other cells, which are these round cells, are the red blood cells in the body. Okay. You can see here that it is continually chasing that bacteria. The bacteria is evading it. Finally, it is going to come and engulf it. So, the suspense is resolved here, right, and it has engulfed the particular bacterial cell, right. And there are multiple things that are happening here, right. So, if you look at it in this way, so you have the neutrophil which has to continually know where the bacteria is. So, the bacteria will shed certain molecules, and that the molecule will set up a gradient, right. So, that the gradient or concentration of the molecule is highest near the bacteria, and then far off from the bacteria it will taper off, right. So, then this neutrophil is a huge cell body compared to the size of the bacteria. So, it will be able to know that, okay, in the front of the neutrophil there is a high concentration, in the back there is a low concentration. So, across the different regions surrounding the neutrophil, it will be able to say where the concentration of the bacteria is the largest and where it is the lowest. So, then based on that it is going to move towards the bacteria, right, and then eventually it engulfs it, and there is so much real time decision the neutrophil has to make in order to properly engulf the bacteria, right. So, any wrong decision we are going to, it is going to be missing the bacteria, and then if you miss too many bacteria, then your infection is not under control in the body. Okay, so another motivation of why we need to or why the body cells need to be constantly monitoring its environment, right. Okay. So, now we come to the actual biological question. So, we have just seen why a cell needs to sense its environment, but then how does it sense it? What are the molecular routes through which a cell senses these external signals and relays it to the nucleus, right. And we are going to first show some cartoons, and then we are going to slowly increase the complexity of all the routes that are possible that the cell can take to sense and relay these external signals. Okay. So, this is a cartoon view which is what we call as a canonical signaling pathway. This is like a very common structure that you see in many living cells. We have the cell membrane here, right. So, I am going to label certain things. So, this is the cell membrane. This is just one example or one representative signaling pathway, right. This molecule that spans this cell membrane is called a receptor. This is basically a protein. It has some part of the protein is sticking outside the cell. So, this is the exterior of the cell. This is the cytoplasm or the interior of the cell, right. And so, this is the cell membrane, right. So, we have the receptor protein, which is essentially a molecule, right, that is sticking some part outside and some part inside. A ligand is essentially a signaling molecule that is in the exterior of the cell. In the case of this neutrophil and bacteria, some of the molecules that are shed by the bacteria, those will be the ligands, right. And so, that will be the input. So, these ligand molecules will then be binding to the receptor and it will activate the receptor. Once it activates the receptor, then this receptor can go on and activate other proteins. So, this MAPKKK is a protein that is within the cell, and that can get activated, and this activation essentially means that there are some phosphorylation happening. So, there are some phosphate groups, these P's refer to these phosphate groups that are added to certain amino acids on this protein MAPKKK, right. So, from this inactive form MAPKKK, this becomes the active form MAPKKK with some phosphate groups added, and these phosphate groups are added whenever the receptor is activated, and then it a kinase is a name given to a protein that adds phosphate groups to other proteins, right. So, this activated receptor becomes a kinase, and or it works with a certain other proteins, and then those other proteins become kinases which activate MAPKKK by adding these phosphate groups, okay, to certain amino acids on these proteins. Once this MAPKKK is activated, then there is a cascade that starts on, okay. So, you may have only like a very few number of ligand molecules outside the cell, maybe tens of copies, but then through this process the signal is going to be amplified, and you may lead to hundreds or thousands of copies downstream, okay. So, maybe this 10 copies of the ligand may lead to 100 copies of MAPKKK proteins that are activated, and these activated MAPKKK proteins themselves become these kinases, and they are written like drawn like pencils here, because sometimes they are also referred to as writers. So, they are essentially writing these they are adding these phosphate groups or writing these phosphates onto some amino acids in the in these proteins that are being activated, okay. So, then there is another protein called MAPKK that is in its inactive form, but once this MAPKKK is activated, then that can in turn activate MAPKK and then add a few more of these phosphate groups, okay. So, this phosphorylates MAPKK. MAPKK can in turn become a kinase and activate MAPK, okay. And once MAPK has been activated, it can then activate a few other proteins, regulator proteins, which can then come and bind in front of the DNA upstream of certain genes. Here there is a red color gene that is being shown, and then it can activate the gene and then produce the protein product for that corresponding gene. That will be the output of this whole signaling pathway, right. So, the input, which is this ligand or signaling molecule, so hormones like insulin are signaling molecules, and in case of neutrophil and these bacteria, whatever molecules the bacteria sheds or those related to the bacteria can be signaling molecules. So, those few copies of the ligand are amplified and relayed through this signaling mechanism and eventually goes inside the nucleus, right. So, this one is the nuclear membrane. It leads to some transcriptional event which is happening within the nuclear membrane, and then the protein that is activating this transcription or regulating this transcriptional event is called a regulator protein or transcription factor. So, this green protein is the transcription factor, right, and that is activating the transcription of this red gene, right. You can see here that this is a canonical signaling pathway, and we can simplify it even further if you want to write it all down in a linear sequence of events, right. So, you have this ligand here which then interacts with the receptor. I am going to call this as the R protein, the receptor protein, and then you have this protein which is this MAP triple K that it activates, and then this MAP triple K activates MAP double K, and this MAP double K activates MAP single K, and that activates some transcription factor. I am going to call it TF, which is this green one here, right, that is your transcription factor or regulator protein, because it regulates the gene expression of another gene which is the red gene. So, then you have the red gene which is the final regulator gene, right. So, this is the another representation of what is shown on the right. Okay. So, the shown on the right is bit more biology being shown. On the left we are showing it as a series of protein interactions, right. So, you have this receptor protein interacting with MAP triple K interacting with MAP double K, and so on. Okay. And that is shown here for a particular example in yeast. So, this is a canonical signaling pathway in yeast involved in mating response in yeast. Like when two yeast cells want to come and fuse together, then there is a certain each of the cells needs to then sense that there is some other cell that it wants to fuse with, right. This is a series of proteins involved in this mating or pheromone response in yeast, and these MAP triple K, MAP double K, MAP K are all these different kinases, and there are specific proteins that can perform the role of these kinases. So, in this particular case in yeast, STE11 or ST11 is performing the role of this MAP triple K. STE7 is a protein that is performing the role of this MAP double K, and Fuse3 is a MAP kinase, okay, MAP K protein. And then there are other genes involved in relaying this signal from the receptor, which is either STE2 or STE3, and then finally you have transcription factors such as or these regulator proteins such as STE12, okay, that are involved in activating the expression of certain downstream genes. Okay. Now, you may ask the question, so that is one canonical signaling pathways, but are these all the known signaling pathways? And the answer is not really, because we have 20,000 genes in our human body, they all encode for 20,000 proteins roughly, right. And so, it ought to be more than just one that is shown here. This is an yeast signaling pathway, but there are some equivalent signaling pathways that are also there in mammalian cells, right. But it cannot be just this one signaling pathways, because a cell needs to sense multiple types of environmental signals, and then it has to act accordingly. So, there has to be definitely a lot more, and then that is what is shown in this diagram. So, there are some signaling pathways which respond to growth factors, and there are others which respond to death factors, right. And then there are some which respond to these chemokines, which are signaling molecules that make a cell move towards a particular direction, right. So, it stimulates cells to migrate towards a certain direction. And then there are some larger class of signaling molecules called cytokines. Okay. So, for instance, here these growth factors come through a receptor called receptor tyrosine kinase RTK, and then you have a series of protein interactions, and then it goes inside the nucleus, and then you control the regulation or expression of certain set of genes. Okay. So, this is your cell membrane, this is your nuclear membrane, and you can see that many of them have these different ways in which the exterior information on cytokines or chemokines or growth factors or death factors, all of them going in and and converging in different parts of the interior of the cell. Okay. But still, this does not capture all of the signaling interactions that are happening within the cell. We may want to discover new modes of signaling interactions, right. So, new modes of signaling pathways, right, and that is really the question. Are there more routes through which a cell senses and relays external signals? And even for those routes that are already established, are there any new players in those signaling pathways? Okay. So, that is what we would like to answer.